But today in this we are going to learn about what is health and what is diseases, and some of the life histories of the parasites which are responsible for causing various types of diseases. So term health is a very frequently used word by everybody, and how do we actually define it? So for a long time, health was actually considered as a state of a body and mind where there was a balance of certain humors, and it's actually called as good humor hypothesis. According to this hypothesis, in the olden days, the health is actually defined as it is a state of body and mind where there was a good humors, and humors means the body fluids. And so this hypothesis was actually asserted by the Hippocrates and the Indian Ayurveda system. So according to this hypothesis, persons who were provided with a black bile, who are actually considered as provided with a hard personality, and would have also the fevers and having the high body temperatures. And so according to this good humor hypothesis. And then later William Harvey, he is actually the scientist who discovered the blood circulation in man, he actually disproved this good humor hypothesis, and he actually defined that the blood is actually responsible for the maintenance of the body temperature, and using with the help of thermometer in the persons, and those who are provided with the black bile, they are not actually provided with the high body temperatures. And so with the help of the thermometer and the blood circulation, he proved that it's not the humors and the body fluids which are actually responsible for the having the hot or the cold personalities. And so what exactly the health is about? And according to that, in the modern view, the health is actually defined as it is a complete state of the physical, mental, and social being, and that's what we actually define in the modern terms. And so health is not actually the absence, a mere absence of the disease, or it's actually not the absence of the physical fitness. And so health is actually about it's a complete physical, mental, and social being. We actually call it that individual is actually considered as the health. And now what are the actually the factors which are actually affects the health? And so there are the sometimes the genetical disorders are present, which are the by birth. The individuals usually suffer from the various kinds of the disorders which disturbs or affects the health. And another one is the infections, and which are actually caused by the various types of the microbes. And the third kind which is responsible for the affecting the health is the way we actually lead our life, and that is the lifestyle, and that is the amount of the food that we eat, type of the food that we are eating, and how we are consuming, and is at all we are get affected with the various kinds of the alcohols or the drugs or the abusing of all those kinds. And so the way you are leading your life, this is also responsible for the affecting of the health. And yeah, when the people are healthy, actually they can able to work more efficiently, and when they started working more efficiently, it is actually responsible for the increasing the productivity, and also it brings the prosperity. And so health is also responsible for the increasing the longevity of the people, and reduces the infant and the mortality rate. And the health is also can be get improved with the help of the balanced diet and the regular exercises. And the very important thing is that doing the regular exercises and the practicing of the yoga. Since the time immemorial, as you know that the yoga has been actually get practiced, and for both the physical actual and the mental health maintenance. And there are the various types of the body functions which actually occur inside our body, and knowing about all the body functions, and what kind of the food that we are eating, and what kind of the infectious agents to which we are get exposed, and leading the good development of the good habits. These are all the knowledge is actually required for a particular person to maintain his health. And so how the health is actually get achieved by it? So taking of the proper vaccination at a time, and the awareness of the various kinds of the diseases, and the another one is the proper disposal of the biological waste, and the vectors which are actually responsible for the spreading of the diseases, and how to control them. And then maintenance of the hygienic food, and so eating of the good food, and the clean water resources, and all these are responsible for the improvement of our health, and all these resources are actually necessary for achieving good health. And now coming to what is exactly the disease is all about. And it is a change from the normal state of the health, which is in turn responsible for the causing of discomfort and the disability inside the body. And so any change inside our body which is responsible for the causing of discomfort and the disability inside our and if you are developed with a disease and which can be get identified with the help of various kinds of the symptoms that are actually get produced as a result of the disease. And so which are you can also consider that the different types of the signs are the symptoms by due to the development of those, we can consider that a person is actually get affected with a so and so particular type of the disease. And infectious diseases are those which are actually get transmitted from the one person to the another person and which are actually caused by pathogens and they are also very common to them. And so diseases which are actually get transmitted from the one person to the another person and which are also caused by the pathogens and which are actually very common to the human beings. And among them, some of the examples are like amoebiasis, dysentery, or the amoebic dysentery, and we can call it as and the malaria, elephantiasis, and the typhoid, pneumonia, common cold, ringworm. All these are the different types of the diseases, the infections, and which comes under the infectious diseases. And under the non-infectious diseases, and so those which are not transmitted from one person to another person and also not caused by the pathogens. And so for example, genetic disorders, kidney problems, heart problems, which are all they actually come under non-infectious diseases. And now let's move on to the some of the different types of the parasites or the common parasites which are responsible for the causing of diseases in a man. So the disease causing forms or the disease causing organisms are called as pathogens, and the pathogens can enter inside our body through the various ways, and various means like sometimes the inoculation or by means of contamination. There are the different ways are there by which the pathogens get entered inside the body and responsible for the causing or development of diseases. Let's move on to the some of the common parasites responsible for causing diseases to a man. Among them, the first one is the Entamoeba Histolytica. It belongs to the phylum Protozoa and the subphylum Sarcomastigophora, class Rhizopodian. So it's one of the very common parasite responsible for the causing of the disease called amoebic dysentery. It's a monogenetic parasite because it completes its life cycle in a single host, especially in case of the man. And where it resides, it actually inhibits the large intestine of the man. So man is actually the single host for this, completes the entire life history of this parasite. Responsible for causing of a disease called amoebiasis or amoebic dysentery in man. The mode of infection is mainly through the contaminated food and water. And so whatever the food that he take and whatever the water that he's in, which is actually contaminated by the stages of this parasite. So in taking of those things, the parasite actually enters inside the man. So houseflies, cockroaches, or some of these insects actually acts as mechanical vectors for this parasite. And mechanical vectors means the stage is actually carried by this parasite, and the parasite do not undergo any kind of the life history inside these insects like houseflies or the cockroaches, and only the stages in the of this parasite being get carried away from the towards this food and the water, and which is actually consumed by the human beings. And it is cosmopolitan in distribution, that is worldwide in distribution, and the parasite is prevalent in almost all the places all over the world, and especially but the more common in case of tropical and the subtropical regions where you will find the more density of the population is usually present, or the unhygienic conditions are present, or the poor sanitation, or the poor people who are present, and highly dense or unhygienic conditions. In all such areas, the parasite is actually the more prevalent, and these are the areas it is actually the more prevalent. Coming to the structure of the parasite, one of the stages in the life history of the parasite, we call it as trophozoite. Trophic means it is the stage that is actually responsible for the feeding, and trophic means feeding stage. So trophozoite stage, it is the most active and the motile feeding stage, and it is also responsible cause the pathogenic stage. And pathogenic means This is a stage which is responsible for the causing of disease, and this is a stage which is moveable with the help of by forming a single pseudopodium. It is also a movable and motile and being active and the metabolically active and where it resides, and it actually lives inside the mucosa and the submucosa inside. As you know that the intestine has got the 4 different layers, and outer serosa, and below that one you will find the muscular layer, and below that one submucosa and mucosa. 4 layers are present in the wall of the intestine or the large intestine. Among that 4 layers, it actually it resides inside the mucosa and submucosa. So, mucosa and submucosa, from the inner side, these layers are present, and inside that layers this parasite is actually present, and it is provided with a single large blunt pseudopodium is present. As the pseudopodium, it is a finger like projection as our small lobe like structure in is present. So, it is called the pseudopodium is actually called as lobopodium. As it is provided with a one pseudopodium, it is called monopodial condition, and the entire structure is surrounded by a plasma membrane or plasma lemma. And whatever the cytoplasm that is present inside this one, which is differentiated into outer and inner, and outer ectoplasm and inner endoplasm. Outer ectoplasm is clearly viscous and non-granular, whereas the endoplasm is fluid like and the more granules are present. And inside this endoplasm, you will have got ribosomes and food vacuoles, and the nucleus is also present. These are all organelles are present inside this endoplasm, and whatever the nucleus that is present, the nucleus is described as vesicular nucleus, and see that one clearly it visible like it is a cartwheel like nucleus. And so, whatever the chromatin material that is present inside, and the endoplasm present inside the nucleus, and the nucleoplasm inside that present, these 3 components of the nucleus are arranged in such a way that the nucleus actually appears as a cartwheel like a nucleus. And this parasite is actually without contractile vacuoles are absent, the absence of the contractile vacuole because it lives in an isotonic medium. And so, whatever the concentration of the tissue fluid present inside that large intestine of the submucosa and mucosa, the concentration of the cytoplasm of this parasite is also the same, and so it lives in isotonic medium. So, there is no need of contractile vacuole to present. Then endoplasmic reticulum is absent, and the Golgi apparatus and mitochondria, these cell organelles are also absent, and as mitochondria absent and it undergoes the anaerobic respiration, it is an obligate anaerobic in nature. And so, absence of the mitochondria actually indicate the obligate anaerobic nature. So, only anaerobic, no oxygen is taken inside and to show the aerobic kind of respiration. So, due to the absence of mitochondria. So, and it is one more important thing is it produces a proteolytic enzyme, and this proteolytic enzyme is called histolysin. Histo means tissue, lysin means breakdown. Whatever this proteolytic enzyme produced by this parasite responsible for the breakdown of the tissues and especially the wall of large intestine which is present in the mucosa and submucosa layers of the gut wall. And what kind of the nutrition it exhibits, and nutrition is actually holozoic, and it's holozoic means completely animal like nutrition. That means it ingests on the solid type of food materials. That kind of the mode of the nutrition we call it as holozoic. And the whole food vacuoles which are present inside, the food vacuoles that are present inside which are provided with an ingested bacterium or which are also provided with an ingested RBCs are present inside. So, these presence of the cartwheel like nucleus and the food vacuoles are provided with the erythrocytes, and these are the some of the characteristic features, the main characteristic features. And so, we can able to differentiate this Entamoeba from amoeba only mainly by these 2 characters, and that is presence of cartwheel like nucleus, that is a one important feature, and food vacuoles which are also provided with a ingested RBC. And these 2 characters from which we can easily identify the Entamoeba from the amoeba. Move on to the another stage in the life history of this parasite, which is the called the pre-cystic stage. So, before the forming the cystic stage, and compared to that of the trophozoic stage, trophozoite is a feeding stage and the pathogenic stage. Whereas the precystic stage, it is a non-pathogenic, it do not responsible for it do not cause any kind of the disease, and it is also non-feeding, it cannot able to ingest on the materials. And where do you find trophozoite found within the wall of large intestine, that is submucosa and mucosa, whereas precystic is present inside the lumen of large intestine. It comes out from the wall and get transforms into this one. And compared to that of the trophozoite and the precystic stage is very smaller and inside and non-motile. Inside that because it do not feed, so it is provided with a reserve food material. And so that reserve food materials are glycogen granules and the chromatoid bodies. The chromatoid bodies are nothing but the ribonucleoproteins and that are present inside that. The only reason presence of the reserve food materials is do not feed on, so which is actually present within the lumen of large intestine. The next stage is the cystic stage, and the stage first one is the trophozoite, then the precystic stage and the cystic stage. The cystic stage, it is also a round in shape which is present within the large intestine, and after the slowly it forms or acquires and the cyst is get developed around this stage and get transforms into a cystic stage. So once after acquiring the cyst, because the cyst is actually secreted by itself, and whatever the precystic stage is present, that secretes and forms a cyst around the whole body. And then it's actually a delicate and highly resistant cyst wall, and that is secreted by this parasite. Now the formation of the cyst around its body is called as encystation, which is means to why it forms the cyst, or why what is necessary for the undergoing encystation. Mainly to tide over the unfavorable conditions, because once the parasite leaves the host and comes out into the external environment, to sustain that all that unfavorable conditions, and so it need to develop the encystance, because the parasite comes out from the body of the host, and to enter into the another host it need to come out from the body of the host and into the external environment. So to tide over that unfavorable conditions, it actually secretes the cystine. So once the it's a transformation of cystic stage is over, inside that one the nucleus which is present, and this nucleus undergoes two successive nuclear divisions. So those two successive nuclear divisions make it to get formed into a tetranucleate stage. And so this is a cyst. Inside the cyst the parasite is present, the nucleus undergoes the two successive mitotic divisions, results in the formation of 4 nuclei. So the 4 nuclei containing a cyst and the parasitic stage, we call it as tetranucleate cystine, or which we also call it as mature cystine. So this stage is in the life history of the parasite is actually considered as the infective stage of a man. Let us move on to the life history. Now we already studied the trophozoite stage, and the trophozoite stage resides inside the mucosa and submucosa of the large intestine, and where this trophozoite keep on feeding, and as it keep on feeding and then it also increases in size and keep on multiplying. So multiplication of the trophozoite occurs in the wall of the large intestine, and it reproduces asexually by means of a binary fission. So it undergoes binary fission and the daughter amoebae are produced. So like that keep on dividing and producing the several number of the daughter amoebae, and these daughter amoebae which are present inside the intestine, and they get transformed into one. So whatever the daughter amoebae that are present, so these daughter amoebae this is a trophozoite stage, and this trophozoite stage multiply by means of a binary fission, so divide into the daughter individuals. And so some of the daughter individuals and again remain within the large intestine wall and become mature trophozoites mature trophozoites, whereas some of them leave the wall of the large intestine and enter into the lumen of large intestine, where they get transformed into precystic stage. We already studied the characters of this precystic stage, and the later precystic stage acquires a cyst to get transformed into the cystic stage, and inside the cystic stage the nucleus undergoes the two successive mitotic divisions to transform into the tetranucleate cystine. So just now we discussed this tetranucleate cyst is the stage that is regarded as the infective stage to man. So, now the tetranucleate cyst which is present inside the large intestine, that comes out along with the fecal matter. So, it comes out along with the fecal matter, and once it comes out out of the host along with the fecal matter to enter into the new host. So, in this external environment of the host, and how long it can able to survive. And so, it almost remain alive for about 10 days. And so, till the suppose within the duration of the 10 days, if they could able to reach the new host, especially by means of the contamination. So, the fecal matter which is contains the tetranucleate cyst contaminates the food and the water, and by chance if it could able to enter into the new host. And so, in such a way it enters inside the a new host along with the contaminated water and food. So, after entering inside that and so it passes through the different parts of the alimentary canal and reaches to the small intestine, where inside the small intestine, after reaching over there, so by the action of the enzyme, the enzyme called trypsin, which is present in case of the pancreatic juice. And so, the trypsin enzyme acts on the wall of the cyst wall. So, that cyst wall dissolves, and from that the stage comes out is called as excystic amoeba or metacystin. And the process of the dissolving the cyst and coming out of that cyst, we call it as excistation. And the parasitic excisted amoeba, it's from one cyst only one metacyst comes out. And now, this metacyst again undergoes the multiplication. So, metacyst undergoes again the multiplication and responds by undergoing the simple binary fissions, and all the nuclear divisions and occurs inside them, and develops into the several number of the increase in their number, and multiplication occurs inside. So, as a result of the multiplication, so 8 daughter Entamoebalae are formed, and so inside this one 4 nuclei actually undergoes the nuclear division, becomes into the 8 nuclei along with the cytoplasm divides to form into 8 daughter Entamoebae or the metacystic trophozoites. And so, whatever these metacystic trophozoites which are formed, and now they reach the wall of the large intestine, and become, after attacking into the large intestine, again they get transformed into trophozoites and continue the life history. And so, they will grow and transform into the mature trophozoites. So, like this they keep on increasing the number by attacking the large intestine. So, sometimes so these trophozoites dissolve the. So, if you look at in sequence, tetranucleate cyst after excistation transforms into the metacystic stage, metacystic trophozoites are formed, and now these trophozoites invade the wall of the large intestine, and where they grow and transforms into the metatrophozoites. And now, how they are responsible for causing of the disease, that what we call it as pathogenicity. So, these trophozoites which are responsible for the production of the histolysins, these histolysins are responsible for the dissolving the wall of large intestine, especially the mucosa and submucosa. So, the release of this histolysin responsible for formation of ulcers in the submucosa of large intestine. And as that the blood vessels and the large intestine wall, submucosa is the layer in the large intestine provided with the blood vessels, nerves and lymphatic vessels are present. So, with this effect of the histolysin, it dissolves the blood vessels present within the wall of submucosa. So, that blood also oozes out, and along with the blood RBCs and which comes out, which are also get ingested by the trophozoite. So, cellular and lymphocytes, red corpuscles, bacteria, all are get ingested by this one. So, cell debris, RBCs, lymphocytes in the ulcers, and that results in the formation of abscess. And so, these some of the stages which are present in the large intestine, they started coming out and along with the stool. So, along with the fecal matter or the stool, the blood comes out and the mucus also comes out, and some of the stages of the parasite like the tetranucleate cyst also comes out. And so, the condition we call it as whatever the condition that is developed, we call it as amoebic dysentery. So, or the intestinal amoebiasis or tropical amoebiasis. And sometimes there are certain individuals which are resistant to this parasite, they are called carriers or asymptomatic cyst carriers. And They do not actually exhibit any kind of symptoms, but they are responsible for the passing of tetranuclease cysts through their stool. So, in which you do not find any symptoms, and such individuals are called carriers, and they are also called as asymptomatic cyst passers. And in extreme cases, sometimes the abscess are also formed other than the large intestine in the various kinds of the organs, and that is outside the alimentary canal also. So, if at all any abscess that are formed as a result of this parasitic infection other than the large intestine, then we call it as secondary amoebiasis. So, sometimes these trophozoites may enter into the blood circulation and from the large intestine. So, because from the large intestine and the small intestine only the blood is actually get collected and get distributed to the different parts of the body through the liver and heart. And so, as the blood is get collected from the large intestine, some of the stages of these trophozoites also go along with that blood circulation. First they primarily go to the liver, and from there they go to the heart, and from there they go to the different parts of the different organs of the body. And so, where they are responsible for the formation of abscess. And other than the liver, other more areas prone for the formation of abscess are lungs, kidneys, brain and heart. And in all these there is a chance of formation of abscess. So, this amoebiasis is called as extraintestinal amoebiasis or secondary amoebiasis. Now, how to prevent this disease but not to develop, and that's measures we call it as prophylactic method. So, it is always better for the prevention is better than cure. So, the prophylactic measures are using boiled and the filtered water, or by the washing hands and the fruits and vegetables that we eat, and using septic tank toilets, and avoiding the urination or the defecation in the open areas. And so, by which you can able to prevent this contamination of the food and water, and also the development of this disease. Now, Plasmodium, it's belong to the phylum Protozoa, subphylum Epicomplexa, previously known as Sporozoa, a class Telosporia, and the genus Plasmodium, and the name of the species is Vivax. So, Plasmodium Vivax, unlike the just now we studied the parasite, Entamoeba is a monogenetic which requires it completes its life cycle in a single host, whereas Plasmodium Vivax, it is a digenetic parasite. It requires the two hosts to complete its life history, and it is the intracellular parasite. And so, it lives inside the cells of the host especially, and like the erythrocytes of the man or the liver cells of the man. So, because of that it is called as intracellular parasite. And whereas it is also completes some of the part of its life history in mosquito, where it actually lives as an extracellular parasite. Out of the two hosts, the man and the female Anopheles mosquito, man is actually considered as a secondary host, female Anopheles mosquito is considered as a primary host. And whereas the monkey is also regarded as the reservoir host. And so, reservoir host is a host where if the primary or the host is actually, or the secondary host is actually not available, in such cases for the chances of to increase the survivability, this parasite survives inside the monkey, where it do not undergo any kind of life cycle, and that is the reservoir host. And unavailability of this secondary host, it enters into the reservoir host, where it survives and it do not undergo any kind of life cycle. Now, the infective stages of this parasite are called sporozoites, and which are sickle shaped structures, and the mode of infection is by means of inoculation, and the mosquito bites are actually responsible for the causing or the infection of this parasite. So, among this Plasmodium parasite which are responsible for causing malaria in man, there are the 4 different types of the species are actually present responsible for causing the different types of malaria. Among them, Plasmodium vivax are responsible for causing benign tertian malaria. It is the most common type of malaria. The second one is a Plasmodium falciparum, responsible for causing malignant tertian malaria. Plasmodium ovale, responsible for causing a mild tertian, and Plasmodium malariae causes the quatern malaria. So these are the 4 types of malaria in man responsible for causing the different types of malaria. Let's move on to the little bit history related to that of the malarial aspects. So Charles Lavron, he is a scientist who actually discovered this parasite inside the blood of a patient suffering from malaria. So Charles is the first scientist who observed the presence of this parasite inside the blood of that patient. And later Sir Patrick Manson actually suggested that this parasite maybe gets spread by means of mosquitoes, because he is a scientist who actually started working on the Ochraria bankruptcy life cycle and suggested that maybe spread by the mosquitoes. So based on his information, Ronald Ross started working on the female Anopheles mosquitoes and discovered some of the stages of this parasite, especially the oocysts of this parasite on the stomach wall. Actually we call it as a crop wall of female Anopheles mosquitoes. So Sir Ronald Ross discovered the oocyst on the stomach wall, or we call it as in insects call it as cropper, on the crop wall of the female Anopheles mosquito. Later Grassi and other scientists also started working the life history of Plasmodium Vivax even in the female Anopheles mosquito. Golgi also discovered this parasite inside the erythrocytes of man. So Golgi, Grassi and Ronald Ross, Sir Patrick Manson, Lavron, they're all these scientists in a sequential form. They all are responsible for the studying of this. They studied and discovered some of the stages, and other subsequent successive scientists actually discovered the whole life histories of these parasites. Now, what exactly the term malaria? In olden days, it is believed that it is actually caused by inhalation of the bad air. So because of that, the name of the disease has given the name malaria. But later it's came to know that to the various kinds of the experiments and all, it's not due to the inhalation of the bad air, it is due to the inoculation of the female Anopheles mosquito bites and responsible for the spreading of this disease and caused by the parasite called malaria. Interestingly, the World Mosquito Day is celebrated on the August 20th, and Malaria Day is on the April 25th. Let's move on to the one of the infective stages of this parasite. So sporozoite are the sickle shaped structure which is actually responsible for the this is a stage in which enters into the body of the man, and that is the secondary host. So the infective stage to the man is the sporozoite. If you look at the structure of the sporozoite, it is totally the ultramicroscopic structure of a sporozoite, and it is entirely surrounded. It's a sickle shaped structure, slightly curved and totally surrounded by a layer called pellicle. Elastic pellicle is present, and within the wall of the pellicle microtubules are also present, and inside that one a highly folded convoluted tubules are also present, whose function is actually not known. When you come to the anterior part of this parasite, it is provided with a apical cup-like structure. A small depression-like apical cup-like structure is present, into which the secretory granules also get opens. So this is the part which helps in actually entering into the body of the host by secreting the cytolytic enzymes. So these secretory granules releases cytolytic enzymes into this apical cup and provide with the apical sucker. So because such kind of the apparatus are provided to enter into the cells of the host, later the subphylum name is changed from Sporozoa to Epicomplexia. Such a presence of a complex structures to helping entering into the cells of the host. So especially this apical cup along with their secretion cytolytic enzymes helps in entering into the liver cells of the man. So this is a simple structure, and inside that one we have got all the cell organelles are present, and like nucleus, Golgi complex and endoplasmic reticulum, all the cell organelles and mitochondria, all the cell organelles we can able to see, and their mitochondria is present, Golgi complex is present, remaining that one like that. But the only thing is convoluted tubule whose function is actually not known. Now let's move on to the life history aspects of the Plasmodium. As we already described, it is a dizentic parasite, completes its life cycle in the two hosts, man and the female Anopheles mosquito. Man is a secondary host, female Anopheles mosquito is a primary host. And why? Because in man it reproduces asexually, whereas in female Anopheles mosquito it reproduces sexually. And now first let us move on to the in case of the life history that of the Plasmodium inside the man. And so in human place, Plasmodium reproduces asexually by means of a multiple fission, which is called schizogony, and it occurs in both liver cells as well as RBC cells. In liver we call it as hepatic schizogony, and in RBC we call it as erythrocytic schizogony. So move on to the each and every one. The one which occurs inside the liver, which is called hepatic schizogony, which is discovered by the Short and Garnham. So now whatever the when a infected mosquito bites the human being, a normal healthy human being, the sporozoites are get released into the blood of a man. Almost nearly in each bite of the infected mosquito releases at about nearly about the 2000 sporozoites into the bloodstream of the man. Now these sporozoites in a duration after circulating inside the blood of man for a duration of the half an hour, and they escape from the blood circulation and started attacking the liver cells. And so in the liver cells, now the sporozoites started undergoing this hepatic schizogony, that is the which is again divided into pre-erythrocytic and exoerythrocytic cycles. And now in the pre-erythrocytic cycle, whatever the sporozoites that enter the liver cells, the sporozoite get transforms into trophozoites by feeding the contents on the liver cells, and then it acquires the spherical shape and attains to the maximum size, which we call it as schizonton. And this is the stage which undergoes a multiple fission, which we call it as schizogony, and is responsible for the formation of the stages that are called cryptozoites. And these we call it as first generation merozoites. Now these cryptozoites which are formed inside the liver cells ruptures the liver cell, and it's and they are get released into the sinusoids. Now pre-erythrocytic cycle, whatever the stages that we studied, and starting from the sporozoite till the formation of the cryptozoites and getting released into these liver sinusoids, it requires about a duration of 8 days, and that is the duration for the pre-erythrocytic cycle. Now later these cryptozoites which are formed released into this one, they may again attack the liver cells to start the fresh exoerythrocytic cycle, or else they may attack the RBC cells to start the erythrocytic cycle. Now in the exoerythrocytic cycle, suppose if they again attack the liver cells, then we call it as exoerythrocytic cycle, which attack the fresh liver cells, and again the same one. These cryptozoites again attack the liver cells and get transforms into again the trophozoites and schizoids, and the same whatever the cycle that is just we saw in the pre-erythrocytic cycle. In the similar manner, the parasite will undergo the same cycle, but this time the stages that are formed at the end of exoerythrocytic cycle are called as metacryptozoites, or second generation merozoites, and that are produced at the end of this cycle. So for this cycle to complete, it only requires the 2 days, and within the 2 days it will complete the life cycle, that is the exoerythrocytic cycle. Now whatever the metacryptomerozoites that are formed as a result of this exoerythrocytic cycle are divided into 2 different types, and some of them are larger in size, some of them are smaller in size. And so those which are larger are called macrometacryptomerozoites, and those which are smaller are called micrometacryptozoites. And so now the macrometacryptozoids attack again the fresh liver cells to continue another exoerythrocytic cycle, whereas the micrometacryptomerozoites they attack the RBC to complete the erythrocytic cycle. So these are the end products that are formed at the end of exoerythrocytic cycle. Then what exactly this pre-patent period is? And so the interval between the first entry of the Plasmodium into the blood of a man and the second entry of the Plasmodium into the blood, and that is the cryptozoites. And So, the stage in which the parasite enters, the first initial entry of the parasite as a result of the mosquito bite, it is sporozoite, and after undergoing the pre-erythrocytic cycle or the exoerythrocytic cycle, especially after the immediately the pre-erythrocytic cycle, only cryptozoites which are liberated into the blood and started attacking the RBC cells. So, the time interval present between the initial entry of the parasite inside the blood of the man and again the reappearance inside the blood of a man, which is called as pre-patent period. So, which is actually approximately about the 8 days. So, during this stage, the parasite during this stage the host body do not show any kind of symptoms. So, there are no clinical symptoms actually, and so it is only the parasite is actually utilizing the body of the host, especially the liver cells, to multiply its number. So, this pre-patent period is wholly useful for the multiplication of parasite. Now, let us move on to the erythrocytic cycle, which was discovered by the which was named after the Golgi, because he is a scientist who discovered this parasite inside the RBC cells. So, that is why it is called as Golgi cycle, and it is this cycle is actually initiated by the two stages, and the stages that are formed as a result of pre-erythrocytic cycle called cryptozoites, the stages that are formed as a result of exoerythrocytic cycle, they are called as micrometacryptozoites. And so, these are the two stages which attack the RBC cells and start the erythrocytic cycle. Now, for example, if the cryptozoites are micrometacryptozoites, they attack the RBC cells to get transforms into a spherical shaped structures called trophozoites. So, trophozoite. Now, this trophozoite inside them, it develops a vacuole inside this parasite and get transforms and pushes the vacuole, pushes the nucleus towards the periphery. So, it transforms into a spherical shaped structure, and inside this one the vacuole develops, and this vacuole pushes the nucleus towards a periphery. As it pushes towards the periphery, it almost appears to the shape of a signet ring like station. So, it forms into a signet ring like stage. Later the vacuole disappears and it develops pseudopodium. The vacuole disappears slowly, and whichever the vacuole, and it develops the pseudopodium, which stage, this stage we call it as amoeboid station. So, later this here the pseudopodium, which is actually formed, not useful for the locomotion, and the purpose of the pseudopodium formation to and also to acquire the amoeboid shape is to feed on the contents of the RBC, especially the hemoglobin. So, it feeds on RBC and increases in its size. Now, we call it as schizoid. That parasite increases in size, it is called schizoid. Now, all these are present inside the RBC. Now, this schizoid undergoes schizogony, that is a multiple fission, and the stages that are actually formed are called erythrocytic merozoites. And almost nearly from inside each RBC cell, nearly at about the 12 to 24 erythrocytic merozoites are formed. Now, erythrocytic merozoites are formed. Let us look at this slide. So, in amoeboid stage, RBCs becomes enlarged. So, as it keep on feeding the contents on the RBC cell size, the parasite increases in size and the RBC cells also increase in size. So, due to the parasitic infection, the RBC cells increase in their size. Such condition is called hypertrophy. Now, whatever it feeds on the hemoglobin, and some of this hemoglobin part, insoluble and disposable hemozoin is formed from the soluble heme part. Now, amoeboid stage, the small red stained Scuffner's dots are also appear inside the cytoplasm of RBC. Now, the hemozoin, the malarial pigment, which is released along with this merozoites. So, the merozoites, as a result of schizogony, erythrocytic merozoites are formed. Along that merozoites, the hemozoin granules are also formed, and which are started releasing into the plasma of the breakage of the RBC cells. So this whole stages are the whatever the stages starting from trophozoite till the formation of the merozoites. This entire the life history inside the RBC get completed in a duration of 48 hours or 2 days. And now the incubation period. Now period between the entry of the plasmodium into the blood and the first appearance or the onset of the symptoms of malaria is called incubation period. So this requires approximately about 10 to 14 days. And so that means it includes you of both the cycles, that is pre-erythrocytic cycle and erythrocytic cycle. And pre-erythrocytic cycle requires a duration of 8 days. Erythrocytic cycle requires a duration of the 2 days. So 8 plus 2 almost nearly the 10 days. And so this pre-erythrocytic cycle gets started by the initial entry of the parasite, and again the onset of the symptoms it actually appear after the completion of erythrocytic cycle due to the release of the hemozoin into the blood. So totally in total or an average 10 to 14 days required for the incubation period. Now whatever the merozoites finally that are formed as a result of erythrocytic cycle, these merozoites keep on again attacking the RBC cells and continue the erythrocytic cycle. So now after the undergoing the several number of the repeated cycles of the erythrocytic cycle, these merozoites after attacking the RBC cells and they get transformed into gametocytes inside the spleen and bone marrow. Now this is after the several number of repetitions of erythrocytic cycle. And so these they without getting transforming into trophozoites, they get transformed into another stage called gametocytes. So whatever the gametocytes that are formed inside the RBC cells which are present in the spleen and bone marrow, they are again divided into two different types, and microgametocytes and macrogametocytes, and which are also called male gametocytes and female gametocytes. And micro they are smaller, macro which are the larger, and the male and female gametocytes. And so now the gametocytes that are formed, the both male and female inside the RBCs which are present inside the spleen and bone marrow, if they are not transferred into the mosquito within a duration of 1 week, the gametocytes will die within the secondary host, that is man. So in order to continue the further life history, they need to get transformed into the another host within a duration of 1 week. And if they are not get transformed, they will die. This slide actually represents the totally the whole life history of this entire the mosquito life cycle. And if just an overlook of the overview of the entire these stages, and this is the pre-erythrocytic cycle. Just see this one, the sporozoite entering into the liver cells, transforming into the trophozoite, then get transforms into the schizoid, undergoes the schizogony and results in the formation of cryptozoites. And these cryptozoites which are released into the liver sinusoids, they attack the they again attack the fresh liver cells and starting the exoerythrocytic cycle. And so again they transforms into trophozoite, schizoid, and some of them are modified into macro larger, some of them are modified into smaller micron. So either these cryptozoites formed from the pre-erythrocytic cycle or the micrometacryptozoites which are formed from the exoerythrocytic cycle attack the RBC cells. And after attacking the RBC cells, see this one, inside the RBC cells the plasmodium parasite transforms spherical, and that stage is called trophozoite. And inside the trophozoite the vacuole develop, and this vacuole pushes the nucleus towards the periphery, and now this almost look like similar to that of a ring like, which is called signet ring stage. And later the vacuole disappears and parasite develops the pseudopodium and appears as an amoeboid stage, and which started feeding on the contents of the hemoglobin present inside the RBC, and that results, and this dissolved heme part and undissolved heme part from that results in the formation of the hemozoin occurs, and also appearance of the Scuffner's dots and inside the cytoplasm of RBC, which is a characteristic feature. Now it increases in size because it feeds on the contents of hemoglobin. That stage is called schizoid. Later it undergoes the schizogony and results in the formation of merozoites. Nearly 12 to 24 merozoites are formed, and those merozoites along with the hemozoin liberated into the plasma and as a result of the entry of this hemozoin, which is being toxic in nature, responsible for causing symptoms of malaria. Now again these gametocytes these merozoites, after attacking erythrocytes repeatedly for several number of times, they get transformed into gametocytes, and see, which are the smaller and spherical in shape, they are called male gametocytes, which are larger, spherical, and which are called female gametocytes. And if these gametocytes are not transferred into the mosquito within a duration of 1 week, they will perish. Life history inside the mosquito phase. So whatever the life history that occurs inside the mosquito of the female Anopheles by this Plasmodium, we call it as Ross cycle, because Ross is the first scientist who discovered the stages of this parasite. So the cycle is named after him. Now gametocytes, and whenever a mosquito bites a malarial patient, it sucks the blood. So whatever the stages in which the parasite present inside the blood, in all that stages it enters inside the mosquito. But out of all that stages, the gametocyte stages are only resistant due to the digestive action of enzymatic action or the digestive action of the crop of female Anopheles mosquito. Remaining all the other stages are get digested except gametocytes. Now inside that mosquito, the gametocytes undergo the stage gametogeny, fertilization, formation of ookinete and oocyst, and the sporosgony. Among them, the first one is actually the gametogeny. So it is a gametogeny means the formation of male and female gametes from gametocytes, and it's called gametogeny. And where it occurs, and it occurs inside the lumen of female Anopheles mosquito crop. Now how the male gametocytes are actually formed. So the nucleus of the microgametocytes divides into 8 nuclei. Whatever the nucleus that is present inside that one, by undergoing mitotic divisions, it divides into 8 nuclei, and the cytoplasm is pushed towards the along with the nucleus to form flagella like structures or processes. And so each pronucleus started entering into the flagella like processes, and that results in the formation of microgamete or male gametocytes. So these male gamete or microgamete exhibit lashing movement and get separated from the male gametocyte. So the process of separation of these male gametes by showing lashing movements from the male gametocyte is called exflagellation. Small slide diagram. See this is the male gametocyte, and the nucleus divides into 8 nuclei and results in the formation of 8 flagella like processes. From these flagella like processes, from the male gametocytes, these flagella like process gets separated, and this process we call it as exflagellation. So like that the male gametes are formed within the lumen of a mosquito. Female gametocytes undergo only few changes and get transformed into female gamete, that is maturation. Then to the female gamete, it will form a small cytoplasmic projection is formed called fertilization cone. So into that, whatever the nucleus that is present inside that one, that nucleus moves towards that periphery into that cytoplasmic fertilization cone at certain points. Just you see that one, this is a female gametocyte. In this female gametocyte, whatever the nucleus that is present, and this nucleus do not undergo any change, but at the cytoplasm forms a small projection like structure called fertilization cone. Now at this fertilization cone only male gamete comes and attaches and makes the female gamete to get fusion. So that union we call it as fertilization. Fertilization occurs, this is also occurs inside the lumen of crop of mosquito. Now the pronuclei of male gamete and the pronuclei of female gamete both are get fused and results in the formation of syncyrion. Syncyrion along with the cytoplasm we call it as zygote. So as male and female gametes are not similar, it is it's like slender, long, and it is a round, spherical, oval shape. So both are not exactly the similar in shapes and the sizes. So fusion of dissimilar gametes we call it as anisogamy. Now formation of the next is ookinete or oocyst. So whatever the zygotes that are formed inside them, so these zygotes later get transforms into a long, slender, motile, vermiform eukaryote, or which we can call it as vermiculum, in a duration of 18 to 24 hours. So it's just change in their shape and becomes long and slender motile form. Now these eukaryotes which are present existing inside the crop, and they started getting penetrating the wall of a crop and get settles under or below the basement membrane of that crop wall, where they becomes round and later secretes a cyst and get transforms into oocystic stage. So now this encysted eukaryote, now we call it as oocyston. Now this oocyston which are present on the stomach wall, so this is a stage in which the Ronald Ross actually discovered this parasite in female Anopheles mosquito. Usually a mosquito infected with this Plasmodium parasite usually provided with 50 to 500 such oocysts are formed on the crop wall. So this is a first time Ronald Ross actually discovered this parasite in this stage. Now this formation of oocyst and the process of the sporogamy which occur on the wall of crop. Now move on to the sporogamy. Formation of sporozoites from this oocyst is called sporogamy. So the nucleus of oocyston which is present undergoes the first reductional division. Now the two pronuclei which are haploid unite together during the fertilization and result in the formation of zygote. So zygote is be diploid, vermicule or eukaryote, it is also diploid, oocyst is also diploid. But before that, whatever the stages that we studied in man and the stages that we are going to study in mosquito, they all are actually haploid. Only the zygote, eukaryote and oocyst, these are the stages in which the parasite exists in diploid state. So being oocyst in diploid, the first nuclear division that occurs is reductional division, later followed by a repeated number of mitotic divisions, and that results in the formation of the next stage in the life history called sporoblasts. So undergoing the first division, reductional division, later undergoing the repeated or the successive number of mitotic divisions was actually discovered by it is according to scientist Bannon. Now after this sporoblasts are formed, whatever the nuclear bits that are formed inside this cyst, they are started surrounding by a little bit of the cytoplasm, and this cytoplasm along with the nucleus getting transforming into a sickle shaped structures and which are called sporozoites. Now oocyst with these sporozoites, now we call it as sporocyston. Now the first one is the oocyst is formed after undergoing all these nuclear divisions and acquiring the shape of sickle shape. Now we call it as sporozoites. Now sporozoites along with this oocyst we call it as sporocyston. So nearly in each sporocyst there are at about 10,000 sporozoites are present, and due to the breakage of the wall of this sporocyst, these sporozoites are liberated into the hemocoel of mosquito. From there they get again travel along with the hemocoel and get attack the salivary glands of mosquito, where they are again ready for the fresh infection to get started. Now the life cycle of Plasmodium in mosquito, it is actually completed about in a duration of 10 to 20 days. Now Plasmodium exhibits an alternation of generations in its life cycle, and Plasmodium vivax responsible for causing the disease called benign tertian malaria. The symptoms actually include bouts of fever which occurs in the three stages, namely cold stage, hot stage and sweating stage. Now cold stage actually the chills and the shivering, headache, giddiness occurs, and in hot stage high temperature of the body, rapid breathing and an increase in the pulse rate. Now in sweating stage profuse sweating and followed by the lowering of body temperature to the normal level. Now as a result of all these, anemia occurs, that is a loss of the number of RBC cells, and due to the more destruction of the RBC cells, spleen also get enlarges. That condition is called splenomegaly, especially in the chronic malarial patients. And In case of repeated number of infections, the spleen also shows the abnormal immune responses enlarges, and which is called tropical splenomegaly syndrome. Sometimes relapse of the malaria and some macrobeta cryptozoites may survive for a longer period in liver, and they remain as dormant stages, which are called hypnozoites. So reactivation of such hypnozoites leads to initiation of fresh erythrocytic cycle, resulting in the new attacks of malaria. This is called as relapse of malaria. So for this malarial disease, the treatment can be cured by a quinine, which is actually extracted from the bark of Cinchona officinalis tree. Out of all these prevention or the treatment and all, prevention is actually the better method. And so the prophylactic measures which are taken are spraying of DDT and BHC, extra insecticides, intervals in the mosquito breeding like places, pools, ponds, ditches and stagnant water, introduction of larvivorous fishes like Gambusia, insectivorous plants like Utricularia into the mosquito breeding places, and avoiding mosquito bite by using nets or the mosquito nets or mosquito repellents, and also by spraying kerosene or pyrethrum oils on the stagnant waters. So if you just look at the overview of the entire life history, see here, parasite sporozoites, as a result of the biting, it attacks the liver cells through the blood, and where parasite reproduces asexually inside the liver cells and the RBC cells in the blood, and reproduces asexually even in the liver as well as the RBC cells, and that results in the formation of gametocytic stages. And these gametocytes again get in this stage, it is entering again into the new host, that is the mosquito, when when a mosquito bites the malarial patient, where these gametocytes are forming into gametes. So after the formation of the male and female gametes inside the lumen of crop of mosquito, and they undergo the fertilization, and after the fertilization they get transformed into the oocysts, and from these oocysts undergo the sporogony, that are results in the formation of sporozoites. And again, when a fresh mosquito bites the again such infected mosquito bites the human being, again it enters into the body of the another man, and repeating the cycle. And in such a way, the mosquito and the man are utilized as a host by this parasite to complete this entire life cycle of Plasmodium parasite.